steps involved in making proteins? Well, before you can build a protein, the instructions must be packaged and transferred from the DNA. First, a specific DNA sequence encoding the protein is transcribed into a complementary strand of messenger RNA in eukaryotic cells. Messenger RNA leaves the nucleus and enters the cytoplasm. In all cells, the messenger RNA binds to a ribosome where the transfer RNA translates the messenger RNA into amino acid. The amino acid or polypeptide chain, which is then folded into a final form as a protein. To understand better this topic, it's necessary to know some concepts. Well, first we have the synthesis, that is the process to combine two objects to to create a new one. We have the transcription that use the DNA as a pattern to build a complementary chain of the messenger RNA. We have complementary that describe two parts that complete each other. We have the translation that decoding short instructions in messenger RNA to build a protein using amino acid. We have correspond that define the relation between a codon of messenger RNA and amino acid.
DNA contains other instructions that a cell needs to maintain itself. These instructions are inside the genes. To be implemented, the instructions contained with the genes must be expressed or copied in a way that the cells can use to produce the proteins needed to sustain life. The instructions stored inside the DNA are read and processed by a cell in two steps: transcription and translation. Each of these steps is a separate chemical process that involves multiple molecules. A codon is a sequence of three nucleotides of DNA or RNA that correspond to a specific amino acid. It is the basic unit of information in the translation process. Each codon encodes an amino acid, and this corresponds is the base of the genetic code that allows to translate the RNA sequence to the amino acid sequence that constitutes the protein. The genetic code describes the relationship between the sequence of DNA bases in a gene and the corresponding sequence of the protein that it encodes. The cell reads the gene sequence in a group of three bases. There are 64 different codons, 61 are specific for amino acids, while the remaining three are used to stop signals. Since there are only 20 different amino acids, there are 6 letters of the alphabet that don't encode a specific amino acid. And the 6 letters will be the B, O, J, U, X, and C. These letters are not abbreviations for any of the amino acids.
Now we are going to see the transcription. First we have that the DNA will start to divide and then the RNA start to form.
In this case we have now the complementary chain of the RNA. Now the letter A and the DNA will change U. Each group of 3 letters will be called a codon. And in each codon will have an amino acid that will form a protein chain.
Now we have a part in where we will see the nucleotides. We have a part in the DNA that are the adenine, thymine, cytosine, guanine. In the case in the RNA, it will change to adenine to uracil, thymine to adenine, cytosine to guanine, and guanine to cytosine.
Okay, as my partner said, in RNA, the adenine changes always to uracil. So here we have an example. It will be adenine, Uracil 1. Uracil adenine U G A G A C G C U A N C.
Now we have an extra part that is in the activity. We have different amino acids that needs to be changed to the correspondent codon. So
These ones are related with the chart of amino acids.
we have this type of table in where we can see the different types of codons that can be formed that are 64 because 4 per 4 we have 64. Then we have like for example the methionine. We will research the methionine in this part of the circle that is in here.
You are always begin with the big letter and then the small one with the very small. So here we are going to have A, U, and G.
Then we have alanine. We will find this in this part. We can see that have different N, is all the letters. So we will take any one to do this. In this case, we will choose J, C, and A. Then we have a part in where we can see that each amino acid had a symbol. So when we see the circle, we saw that each codon will form a type of amino acid and it have a symbol. Then in this part of the activity, we have different codons that we will change to the messenger RNA, and then we are going to determine the symbols that will code a message. So we are going to see the circle to determine this. So in the first part, we have ACU. We are going to find in the circle the A, the C, and the U. We have that is threonine, and we are going to research this in the table of the symbols is with the letter T. Then we have GCU. So we are going to go to this part, GCU, that is alanine. In the case of the symbol, is the letter A.
To describe the DNA sequence to messenger RNA, we use the transcription of the letters, which are A passed to the U, T passed to the A, C passed to the G, and G passed to the C.
If we have a DNA sequence and then we change part of the sequence for another group of codons, the initial sequence will change completely, since they are different amino acids that make it up. For example, we have the next sequence, which forms the next messenger RNA, and this forms the next message. But if we change the first 4 codons, the DNA sequence will be this, and then the messenger RNA will change too. Finally, the messenger will be this.
If we make two different amino acids, the translation of coded message will not be accurate. Also, the protein synthesis will not have the same composition. Now, during the production of proteins before RNA changes leaving the nucleus, the RNA goes to sites of protein synthesis in the cytoplasm.
Knowing how the letters ACGT are transcribed to the messenger RNA, we can transcribe our own DNA sequence. Now we have our own DNA sequence, which is CCT, CTT, TTG, CTC, TJA, TTG, ACG, and AGT. Now, if we change the DNA sequence to messenger RNA, this will be as follows: GGA, GAE, AAC, GAG, ACU, AAC, UGC, UCA. If you can see the messenger RNA, you can see that each codon forms a different amino acid. So in the first codon, the amino acid is glycine. The second is glutamic acid. The third is asparagine. The fourth is glutamic acid. The fifth is threonine. The sixth is isoleucine. The seventh is cysteine. And the last is serine. Now, if we represent each amino acid by the capital letter that was assigned to it, the message will say genetics.